In this video, we will continue our discussion about this IMPDH2, and in this video, we will discuss that IMPDH2 acts as a sensor and localizes to the nucleus due to high nucleotide demand that usually occurs whenever there is a DNA damage repair occurs. So, how they come up to these conclusions? Some of these hypothesis comes from their previous paper where they have did the metabolic analysis of the cell lines that are more prone to DNA damage, and they are able to find out that whenever those cell lines are removed from adipocytes, adipocytes is cause DNA damage, and transferred to the adipocytes free media, then DNA damage starts to occur. And at that point of time, they have observed that there is an increase in the IMPDH2 during the DNA damage repairs. So they wanted to see whether this IMPDH2. So now they go one step further to find out whether this IMPDH2 act as a sensor. So first they replicate their previous results using Western blot, and previously they published the metabolic analysis. So here they did the Western blot where they treat the this triple negative cancer cell lines for a control with DMSO, and for these adipocytes to induce DNA damage, and they can see that. And these Western blots are done after the cells are removed from adipocytes and moved into the adipocyte free media, and let it remain in the adipocyte free media for 24 hours. So, because when they are moved into the adipocyte free media, then DNA damage repair will starts to occur. So, and DMSO also follows the same conditions. Only the difference is that instead of adipocytes, they use DMSO. And when they check the cytochrome, IMPDH2 signals in the cytochrome, they are able to find out that IMPDH2 signals are higher in the adipocyte treated cells compared to the DMSO treated cells. So here it seems like their hypothesis of that IMPDH2 is move towards the nucleus, it seems to be correct, as they do not see any increase in the IMPDH2 or any significant increase in the IMPDH2 in the chromosome compared to the cytochrome. We can see slight increase, but the increase in the cytochrome is more compared to the increase in the chromosome. And once now they have made a hypothesis, there is an increase in the IMPDH2 because of the then because of the DNA damage repair. So in order to link these two lines, in these two sentence, what they have assumed or hypothesized that IMPDH2 act will move towards nucleus because there is a high nucleotide demand occurs in the nucleus. So if in order to validate those hypotheses, what they did is they artificially induce this nucleotide during a DNA damage repair, and this might slow down this IMPDH2 in the transfer IMPDH2 localizations into the nucleus. So this panel B reflects the same results. That is, when they induce guanosine in the DMSO free media, they see that IMPDH2 demands decreases, and similarly, this when they use the induce this guanosine in the ETO, then also this IMPDH2 demands decreases. And thereafter, it seems like their hypothesis seems to be correct, that IMPDH2 localizes into the nucleus because of the high nucleotide demand. And now they wanted to do a dose dependent study and at different time intervals. So they check these IMPDH2 demands. So they induce adipocyte at different concentrations, that is 1 micromolar, 2.5 micromolar, 5 micromolar, 10 micromolar, and check the IMPDH2 at different time intervals after these cells are being removed from adipocyte and keep into the adipocyte free media. And they can see that there is a dose dependent increase in the IMPDH2, both in terms of the increase in the adipocytes cells treated with increase in the adipocytes, at the same time with increase in the time. And these things, immunofluorescence image are also shown in the panel D, where the adipocytes for 10 macromolar clearly shows increase in the IMPDH2 compared to the DMSO treated. And once they validated their results in the triple negative cancer cell lines, they also did their similar studies on the Cal51 cell lines, where they find the similar results. That is, here they check the DNA damage markers using YH2AX, and they can see that adipocyte treated cells has more DNA damage compared to the DMSO control. And those DNA damage will increase with the dose dependent increase in the adipocytes, and at the same time with increase in the time that cells are increase in the times that uh, cells are left it for this DNA damage repair. That is, for example, this for 24 hours we can see that 10 macromolar has more DNA damage, 10 macromolar and 5 macromolar has more DNA damage compared to the 1 micromolar or this 2.5 macromolar. So DNA damage is still working at this 24 hours of time period, because the signals for the DNA damage is still higher at this point of time. So overall, what they conclude from this experimental results, that IMPDH2 localizes into the nucleus because of the high nucleotide demand, and the high nucleotide demands are created in this case is because of the DNA damage repair. I hope you understood their main uh, main conclusions that they come up from this experimental results. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.